Okay, let's go through the nuts and bolts of genetic engineering. Let's move forward. What I want to do is take you through a story, a historical story in the engineering of one of the first drugs. And this was one of the first drugs that was used to treat a human lethal disease. And that disease is hemophilia. And it is a lethal disease. There's absolutely no question about it. People do bleed to death from a bloody nose. I had one this morning and thought I was going to bleed to death, but fortunately it stopped. The point here is that, because my ice maker was broken, so I didn't have any ice. The point here is that individuals with hemophilia, it's a pretty darn serious disease. And back in the olden days, of which a lot of people, New Age people, make so glamorous, people died from hemophilia. And it's a disease in which, probably from a genetic point of view, it's one of the oldest known genetic diseases. A treatment in modern days for something like this is pretty significant because one of the only ways that we used to be able to treat this disease was to give people blood transfusions from compatible normal individuals, because the compatible normal individuals had clotting factors which compensate for the defect in the hemophilia, which is a defect in a clotting factor. And I'm going to talk about that in a second. So this is an adventure that I'm going to take you on of how one goes from a disease to a gene to a drug along the way. And along this way, I'm going to talk about things that you know, and I'm going to talk about things that you don't know, and I'm going to put them all together. Hopefully I'll finish today. If I don't, I'll finish on Thursday. So here's hemophilia. When you cut yourself, you get a scab, and it forms pretty quickly. And so you have a process in your cells which allows a wound to be healed pretty quickly. And what that process is that the wound sets up a cascade. The wound sets up a cascade. Okay. So let's talk about the genetics of hemophilia. So the first thing is, as I've said, it's a disease that goes back to a long time ago, and people recognize that it was inherited because of being transmitted in families, because they recognize it because of circumcisions and people dying. And you can imagine it must have been a very devastating disease, particularly if you're a mom and you had a child and he fell, and died because there was no way to heal the wound. And so what you can see here is, and I'll go into this in a second, is that we're able to do this because you can produce these little fibrins, actually little fibers, at the site of the wound, and those fibers cause the wound to heal and prevent the blood from flowing. And the way this happens is actually a signaling response. Signal, like sending off a signal. And that signaling response is induced by the wound, obviously. The wound, whatever you do, it breaks the skin. And once you break the skin, it breaks the vessels. And it sends a signal for the blood cells to start doing things. And the blood cells have certain proteins in them. And those proteins start what is called a cascade of events. One protein changes another protein, 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 so that eventually you get these fibers, and those fibers are the little, scab that forms on the cut. And they're fibers that are made up this protein called fibrin. And so here's a cascade. And imagine that before the signal of the wound, Hiromi, these proteins have to be inactive. You'd be in pretty bad shape if you started clotting everything in your circulatory system, wouldn't you? Yeah. Did any of you ever read the Andromeda strain? Remember in the Andromeda strain, they basically clotted their bloods. It was a virus from another planet. But the point here is that if you can't form these clots, you're in bad shape. But if, in fact, you have these proteins active in the wrong time, you'll get clots in the wrong place. And so this is a highly regulated process whereby the wound starts taking inactive proteins to active. That active protein takes inactive to active, inactive to active, inactive to active, inactive to active. All the pinks are active. All the non-pinks are inactive. It's a cascade. So eventually, you get this fiber, and that fiber makes the clot. It makes the scab, and the wound is healed. And here's all the genes that are involved, because all of these are different proteins. And if you have a defect in the gene that specifies any one of these proteins, Derek, you have hemophilia. So there's different forms of hemophilia. And each hemophilia gene encodes a particular kind of clotting factor. So factor 8, which is the one that I'm going to talk about, is right over here in this little cascade. Hemophilia B is in this particular factor in that cascade, and there's a whole bunch of others. And when you talk about genetic engineering in goats, you're going to talk about protein C in terms of making that as a drug in goats and in animals. But the point here is that when you have a mutation in any of the genes that codes for any of these factors, Max, you're in bad shape. Do you think that this has to be a regulated process?
Definitely.
What if the inactive proteins were activated somehow by accident?
I would not have circulating blood.
You have a problem.
Yeah.
Do you think that there's other proteins in your cell that keep these inactive proteins inactive in your non-wounded state?
Yeah.
Yeah. And so the point here is that you also have genes that encode proteins that keep these things in an inactive state. And the things that keep them in the inactive state are inactivated on the wound, so there's just a cause and effect and everything is interrelated. Max, what's your hypothesis for if you have a mutation in a gene that no longer can keep this thing inactive?
You would have no real blood.
Well, you'd start clotting abnormally.
Yeah.
Well, that happens. There's this thing called antithrombin deficiency. And people have clotting in their bloodstream when they're not supposed to have clotting. And I can assure you that's a pretty devastating disease because it can lead to a lot of heart attacks and a lot of and premature death. So all of these things have consequences. But the point here is that this is very balanced. It has to be highly regulated. Now, hemophilia of the kind we're going to talk about, hemophilia A and hemophilia B, is on the X chromosome. It's sex-linked. It's on the X chromosome. It's called sex-linked because it's passed down with a certain pattern of inheritance. Mainly, I'll show you how that happens from mothers to sons, and it occurs in about 1 in 10,000 males. It occurs much more frequently in males than females because, in fact, it's sex-linked. And here is the royal pedigree, which we've talked about before. And from this pedigree, you can work out the mode of inheritance. And after Mendel's laws were discovered, people knew that there was a gene in the royal family of Europe which caused hemophilia A. And they came up with the idea that the mutation must be on a chromosome that's involved in sex. And that's what's meant by sex-linked. The gene is on a chromosome which is involved in sex determination. It's not on one of the autosomes. And furthermore, there's a definite pattern here in which you can see that this Beatrice, Queen Beatrice, is a carrier for this gene. And notice that it's passed on to more than half of her sons. And that is a characteristic of this sex-linked inheritance. And the reason why the sons get it and the daughters don't is because the sons, as you know, have one X chromosome and one Y chromosome. So that means in a male, any mutation on the X chromosome will be manifested because there's no other chromosome to mask it. So you don't have in males dominance or recessive relative to the X chromosome. You just have one shot. And if that one shot is a mutation in a deleterious disease, you're going to develop that particular disease. And so as we know, and I'm just reviewing this, that we have sex determination in humans. Females have 2 X chromosomes. Therefore, someone can be a carrier and not have the disease. But in males, there's only one shot here. 1 X chromosome, any deleterious mutation on that one chromosome, it's it. That disease will be manifest. The point is called hemizygous, but I'm not going to get into genetic terms. And furthermore, you can see here that there's a Y chromosome. and an X chromosome, and you know that the reason why the male is because of this SRY gene. Marvin, give me a hypothesis based on what we talked about last week in lecture for how the SRY gene can work. It's a regulatory gene. It specifies one of these transcription factors. It's like the I gene. How does it work to cause maleness?
The SRY gene codes for, is transcribed into a certain mRNA sequence,
Okay.
which in turn translates into different proteins.
Different proteins?
Or certain proteins.
Certain, one gene?
Or one protein.
Okay. And as I just told you, that protein's a regulatory protein. How can that one protein cause these cells to go and form a testis instead of an ovary?
It regulates the...
Tell me exactly, because we covered this in class. We covered it in terms of networks. We covered it in terms of how the I gene works. Covered it in terms of engineering. How does it work? I know that you studied all weekend the notes from last week.
Of course.
I know you stayed up all night studying those notes. So how does it work?
It regulates other genes.
Yeah, exactly. And those other genes are just random genes that are active in the liver, or are those other genes that are forming in the sex-determining cells of the embryo?
There are specific genes that are sex-determining genes.
So they're only active in certain places of the embryo, right?
Right.
Are they active where the head would form in the embryo?
No.
No, they're active where the gonads would form in the embryo. That's exactly correct. So the point is that there's a cascade. These are regulatory genes. Samantha, can I have an XY individual and still be a female?
Absolutely.
Why? Give me several reasons why that can happen, from what you know about genes to proteins.
Well, if you have maybe possibly a deletion or some kind
Of what?
The sequence encoding for the SRY
So the whole gene is deleted.
The whole gene's deleted.
Okay, give me another explanation.
Something's inserted in the SRY gene.
Something's inserted. That's a second. Give me another explanation.
There's a translocation of the SRY gene. It's not in the actual...
I don't know what translocations are, but that's okay. Give me another explanation. So we have an insertion. We have a deletion. What else could happen? More simple than that. Much simpler than that. Brian?
You could have the change of one or multiple
Yeah,
base
one
pairs.
nucleotide. One or more nucleotides. Just a point mutation. You don't have to drastically bombard that gene. So it's possible to have an XY female. And what would be your hypothesis, Tiger, today's your day, is if we had a mutated SRY protein, and since it's a regulatory protein, what won't it bind to on a gene that it's supposed to regulate? Think about the anatomy of genes. What's in the beginning of a gene?
The promoter?
What's before the promoter? Remember we talked about it last time. These, what are they? Anyone? Julie?
The activator.
Yeah, the activator sequences. So if you mutate the SRY protein, it's not going to bind to the activator sequence, and therefore it's not going to set off this process. Do you buy that, Mike? Pretty neat. Okay, so in the course of this disease, hemophilia, we have a mutation in one of the genes on the X chromosome, and it happens to be right at this tip here. And when that happens, you can follow the inheritance. Let's say the mutation occurred here in this woman. It happened in the formation of her eggs. When does that happen, Brian? In DNA replication or in transcription? When does the mutation occur?
Transcription.
The mutation occurs in transcription. How is it inherited from generation to generation if it occurs during transcription?
Oh, I'm sorry. It's replication.
You're positive.
Yes.
Yeah, it has to occur during DNA replication to be inherited. And so this individual is a carrier because only one of the X chromosomes was mutated. And then in passing it on to the eggs and combining with the sperm, we by chance get this male that happens to be a hemophiliac because it has the copy of the gene that was mutated. And so now let's go. Click. Mark. Get set. Eeny, meeny, miny, mo, start. Hemophilia A disease alleles can arise because of change in a base pair sequence, addition of one or more, a deletion, all of the above. 5, 4, 3, 2, 1, stop. display. Excellent. All of the above. It's exactly. Hemophilia A disease allele resides at the same X chromosome locus as the normal wild type alternative. Okay? So that means start, hide. Hemophilia A disease allele resides at the same X chromosome locus as the normal. Yes, no. 1, 2, 3, 4, 5, 6, 7, 8. Stop. Display. Yeah, same chromosomal locus. It's alternative forms, just like the things that I showed you for the tomato. Okay? Let me see if I have another one here. The XY individual is always a male. We already answered that, and let's move on. Okay, so now we have factor 8. We have a human chromosome. Notice that there's these bands on this chromosome, and these bands are markers, and you're going to talk about that in discussion tomorrow. Each chromosome has a unique size and a unique banding pattern. And I'm not going to go into why the banding patterns are banding patterns, but Lauren, what do you think would cause your hypothesis for the banding pattern of the X chromosome to be different than the banding pattern of chromosome 3?
Different.
Dora, do you listening? Go ahead.
Different sequences in the bases?
Yeah, do you agree, Dora? Did you tell Dora? Can you tell her?
I didn't hear what she said.
Here it's coming somehow. If we can get this microphone going. Aren't we giving them the microphones on? Yeah.
Different sequences on the different chromosomes.
Yeah, so do you agree with that, Dora? That
Yes.
these little markers that are these big bands are due to different sequences of the chromosomes?
Yes.
Yeah, absolutely. So we can characterize the X chromosome by its unique banding pattern and by its unique size. Let's continue with this game. Stephen, what's your hypothesis for how someone knew that the factor 8 gene was located right here on that chromosome? How would one have known that? What do you think?
You can test the rates between crossing over.
Don't get simple. I don't know what crossing over is. I don't have a clue. What would be a simple way of knowing that this gene is on that tip of the chromosome?
By sequencing the chromosome.
Can't sequence. Way before sequencing was invented.
By testing for various rates of mutation if the...
In humans, you're going to test for various rates of mutations? How would you know that the gene is on this part of the chromosome? What marks the chromosome, Eden? You're looking at it. How do you know that's the X chromosome? How do you know that's not chromosome number 21?
Because you know all the genes on it.
I don't know anything about the genes on this chromosome.
I'm sorry, I'm not quite understanding what
Eric,
you're
how do you
asking.
know that the chromosome is a unique chromosome?
How do you know the
Yeah, how do I know that's the X chromosome?
characteristics on the X chromosomes are specific to girls, maybe?
Yeah, what might those characteristics be? Do you see anything funny on these chromosomes? Do they look? What are the characteristics of the chromosome, Eric? How can you tell which is 1, 2, 3, 4, 5, 6,
they
7?
all have unique phenotypes.
They have unique phenotypes. What makes them unique? What makes them unique? 23andMe, my butt. What makes them unique?
They produce different proteins.
Eric, look at that picture. What makes them unique?
Size.
Do you know what protein? Do you think that people knew about chromosomes before they knew about sequences and proteins and genes and all these things?
No.
No, I think so.
Or yes.
So how do you determine? I didn't want to digress on this, sort of giving you a lead into your discussion tomorrow. How would you know that that was the X chromosome? Well, you know by its size, because it has a different size than the other chromosomes. It has different size. You can distinguish it from the X. And it has these little bands which distinguish them. And these bands are due to different sequences. And you'll get into what this is tomorrow. And how do you know that the going back to the original question, Chris, why don't you stab at this? How would you know that the hemophilia gene is at the tip of the chromosome? What could happen? What kinds of other mutations did we talk about that involve big changes in chromosomes?
Like whole chunks of chromosomes?
Yeah, what happens if a whole chunk of this chromosome falls out? What might be one of the phenotypes of the individual that might inherit that chromosome?
They would have hemophilia.
And a bunch of other diseases. So could you see the chunk off in the microscope? Would you see it gone?
Yes.
Yeah, absolutely. So the way in which some of these genes were mapped originally is that they had a hunk of a chromosome fall off or a change in the structure, and they correlated that with the disease. And that's how originally it was known that that thing is right at the tip. Okay, let me move forward. So there's the inheritance of the hemophilia gene. I'm not going to go into it. It's sex-linked. You can look at that. But if you look at those chromosomes and how they're distributed in this process of gamete formation, you notice that the Y chromosome goes into one sperm, the X chromosome into another. These two X chromosomes go into two different eggs. Notice one of the eggs has the hemophilia mutant gene because the mother was a carrier. And notice on fertilization, Katie, do you think this fertilization process is random? Do you think that the sperm knows what the egg is?
Does the sperm know what the egg is?
Fertilization, combining sperms and egg, do you think that in that process the sperm can recognize a specific egg with a specific gene?
No.
It's random, right?
Yes.
Yeah. So we're going to have all these different combinations occur. We're going to have a combination of two X's coming together. These are the zygotes. We're going to have a son, that's a female. We're going to have a son that's normal. We're going to have the hemophilia because that X chromosome is inherited with that Y, and we're going to have a carrier. So that means sex-linked inheritance has a very specific type of inheritance pattern, mostly in males and half the males of carriers. And this is how one can use pedigrees to determine what the gene is, whether it's sex-linked, autosomal or not. Faria, do you understand that? Do you see that? Okay, great.
Yes.
So the question then is, here's the $64,000 question. If we have a disease, and we know the disease is lethal, and we know it's devastating, and we've now invented genetic engineering and being able to clone human DNA, how do we find the gene or the messenger RNA and be able to make a drug? Particularly when we don't even know where the protein is made. We know that the factor VIII is a factor. We know it's a protein. We know it's in the blood. But there's a lot of stuff in the blood that's not made in blood cells. So we don't know where it's made. So Kathy, that presents a real problem. Because someone's iPhone just went off. Because if, was that yours, Marvin? Because if you know the insulin protein is made in the insulin, in the pancreas rather, you can go to the pancreas and clone the insulin messenger RNA, right? But if you didn't know where insulin was made, you wouldn't know where to start, would you? In terms of making a cDNA. You with me, Kathy? Sure? So Lauren, if I don't know anything about where the gene is active, what can I start with to get the gene? To clone it. I can't clone the message because I don't know where the message is. What can I start with that I always know is there? I know that it's in every cell of your body.
The DNA? Just like
the gene.
The gene.
Yeah, you can't start with the messenger RNA because you don't know where it's produced. Dora, do you understand that? So if you don't know where the gene is active, you have to start with what you know exists, and that is the gene. It's there. You know it's there because it changes into a hemophilia form. You know it's inherited. You know it's in the chromosomes. You know it's on the X chromosome. You know it's passed on from every generation. So you can start with the gene. If you start with the gene, what kind of library do you make, Anna? Do you make a cDNA library or a genomic library? Library means you make a collection of clones. Genomic or cDNA?
cDNA?
You sure? What's cDNA made from? Made from what? Go ahead.
The mRNA?
So, but if I don't know where the mRNA is, how can I start with that? If I don't know where in your body this mRNA is, I can't grind you up and start with the mRNA, can I? What am I going to start with, Derek? Where's the only place I can start to clone what I want to eventually make a drug?
The chromosome?
Yeah, the chromosomes, the gene. So in order to get the factor VIII to make a drug, you don't start with the messenger RNA like you did with insulin. You have to start with the gene because you know that the gene is there. That means you have to use procedures like making recombinant clones from DNA, just like Article 1 of your discussion. That Article 1, you dealt with making genome libraries because the foreign DNA came from the genome of another organism. And that was used to be put into the plasmids that were present in E. coli. So the question here is, how do you clone a gene when you don't really know very much about it? Okay, let me go on with this. Who haven't I ever talked to? Let me see. Taylor, haven't talked to you in a long time. Let's say that I know a little bit about this hemophilia. I know that it's a defect in a gene. I know that it's a defect in a protein. And let's just say I know that the protein is in the blood. And let's say that I isolated some of the protein from the blood and I was able to get a little bit of a sequence of the protein. How can I find the gene if I have a genome library? How can I go from the sequence of the protein to the sequence of the gene? What do you think? What's this thing called the genetic code? Remember we said if we had the genetic code, you can go from the protein to the sequence of the gene, maybe. What do you think? Am I stumping you?
Yeah.
Okay. Ben, what do you think? Yeah.
Could you repeat the question, please?
Yeah, if I just know a little bit about the sequence of the protein, how can I find the gene if I have a library of the genome? I know the genetic code. I know the amino acid sequence of the protein. How can I reconstruct the sequence of the gene?
You could.
What is a genetic code? It's like a dictionary. What could you do?
You could randomly make a, what's that called, a library?
We already have the library. How do you find the gene in the library?
I mean, you could randomly recombine them and find.
Recombine what? I don't know what one is and.
You could take a sample of the entire genome, cut it up, and recombine it with plasmids and see.
I already gave you that. How do you find the hemophilia gene? I'm going to tell you, but I want to know if you can anticipate it. Dora, do you know? Take a stab. Let's say I have a little bit of the protein that specifies this clotting factor. I know what its sequence is, amino acid 1, 2, 3, and 4. I know what the genetic code is. I give you a library made, let's say, in plasmids or something else, and I want to find that specific gene. How would I do that?
You insert it.
No, you've already done all that stuff. It's okay. It's all new. I know. There's nothing wrong with not putting it all together. It's perfectly understandable. But what it means is that you have to go back and read the article for discussion number 2 when it discusses how you find specific clones in libraries. And it also means that you have to read chapter 2 and 3 of your book because it goes into this. And I know it goes into it because I've had discussions with some of you in office hours that have shown me and have been confused by it, and we've gone over it. So that means you've got to do it. So let me go forward, Eden, and tell you how it's done. So how do you clone the gene when you don't know anything about it? And that's why I'm giving you this review, because I know these are new concepts and these are difficult. So what they knew about hemophilia was that it was X-linked. They knew it was inherited on the X chromosome. They knew it was in the blood. They knew it was a protein. And they were able to isolate a little bit of it from many hundreds of liters of human blood. And they were able to isolate it also from other mammals like cows and pigs. And from a very tiny amount of protein, way back when sequencing proteins was almost impossible to do, they were able to get a small stretch of the sequence of that particular protein, which in those days was a tour de force. Not the whole protein, because it's a gigantic protein, as you'll see, but a small little protein. And so that's what they knew. And so the question then is, how do you go from the protein to the gene? Well, you learned in discussion number 2 how to identify the insulin messenger RNA in a library of pancreas cDNA. Is that correct, Aicha? Am I wrong about that? So you knew how to identify a specific clone in a cDNA library, and it was done by using a probe, something that had a radioactivity on it, and it was done by making a pancreas library from the messenger RNA, making a copy of that library using all the things you talked about in discussion, reverse transcriptase, et cetera, and then you were able to select that clone in a few ways, one of which was taking a radioactive probe of a purified insulin message and screening the library to find the right clone, because in that screening process, that probe formed a double helix with its complementary pairs, and you were able to use radioactivity in order to detect it. Anna, you remember that vaguely? Just, it's okay. It's fine. You're going to get it, because 2 weeks from now, all of you are going to go through this absolutely from one end to the other, up and down the tree. It's pretty simple stuff, as you'll see. This is going to be, by the end of this quarter, this is going to be like walking from here to Ackerman Union. It's going to be so easy for you. So the point is that you can go from the DNA, and you did it. You took a DNA sequence, you put it into a program. The program used the genetic code, and it translated the protein, and you found out what the protein was. So you can clone a gene, you can sequence it, and then identify what the protein is. But what if you know what the protein is, but you don't know what the gene is? Well, you can go backwards. Because if you know the sequence of the protein and the genetic code, you can predict what the sequence of the gene is. You can't get it identical because the code is degenerate, but you can predict it. And if you can do that, you can make a synthetic probe. Synthetic. You make it organically in a lab, one nucleotide at a time. And now you're synthesizing what you think is the sequence of the gene from the sequence of the protein. Remember collinearity, gene sequence, messenger RNA sequence, amino acid sequence, the genetic code. If you know that, there's very specific predictions. So the point here is that we can go from the DNA to the protein by translating it with computer programs after we've sequenced it, after we've cloned it. And we can go from the protein, because we know a little bit about its sequence, using the genetic code to go back and predict the sequence of the protein. So if you don't know where the gene is active, you start with the gene. And that's where this adventure in Factor VIII started with. Started with the protein, started with the gene, started with the protein, started with the gene, went to the mRNA, and went to the drug. Let me digress. What Daisy and Aicha and Kelly and I are doing is we're building a kit step by step by step in genetic engineering. We started the first week in simple plasmids and DNA. The second week, plasmids and cDNA. In between, we talked about properties of genes, what genes do, how they function, talk about how we put them together. Now we're bringing it, as I said, all together as a conceptual unit. Which means, Anna, do you think Michael Phelps won all those medals in the Olympics without doing any work?
No.
This ain't going to come together without doing any work either, trust me. You're going to have to do some work in order to learn this stuff. And that's the bottom line. And when you do, you're going to go, oh, you're going to see it all together. So the point here is, and I'm repeating myself, and I'm not going to go over this, so you can go from the protein to the gene because you know the genetic code. You can go from the gene to the protein because of a variety of things. And here's essentially how you do this. So let me march you through this, and I'll pick up on it on Thursday. So the first thing in getting any gene, if you're starting with the gene, is you have to make a library of the genome. Albert, how do you think they sequenced the human genome? You think they just got the DNA out of the chromosomes and started sequencing, or do you think they first cloned all those sequences?
I think they cloned everything
Yeah,
first.
exactly, because in order to get a specific sequence, you have to have a specific clone. So in order to get the hemophilia gene, you have to clone the genome. The genome, not the cDNA, the DNA in the nucleus. Doesn't matter what cell it is. It could be sperm cells. It could be whatever, but you have to clone it. OK, Mike, in order to clone something, tell me what you need. What are the tools?
You need to have, well, you're going to do PCR. And so
we're going to use PCR?
No, that's the process, yeah.
How are you going to clone the whole genome using PCR when you don't know anything about the sequences? Remember I said in order to do PCR, you need specific primers and a sequence? PCR comes after you clone the genome and sequence it, not before. So what do you need? What are the tools, Brian? Start listing them off. I know you went through this the first 2 weeks of discussion. And I know you guys did that up with Ryan as well. What are the tools? Number one.
An ORI.
In what?
In
what? What do you need?
Well, I guess first we probably want to use a restriction enzyme.
Well, what are you going to need before all that? Give me all the tools. Number one. Do you need any cells?
I do need cells.
What cells do you need? What? Okay, you need human cells. What other cells do you need?
Bacteria cells.
Thank you. So we need cells. What else do you need? Marvin.
You also need...
What?
I don't know. I don't know.
You do know. Don't say that. Okay, come on, guys. I'm doing this on purpose. I could have stood up here, honestly, and I would have been done with this lecture an hour ago. What I'm trying to do is integrate all of these things for you. What I'm trying to do is get you to understand what's important in genetic engineering and what's not, and bring it all together. What I'm also trying to do is prepare you to learn to think on your feet and learn all of this material, because it's pretty simple. And so one has to start preparing because, again, I don't care about the take-home test or any of that stuff. I want you to be able to think about this stuff. Got to do some work. Kelly, do I do any work preparing these lectures? Yeah. You think I'm just up here doing this randomly? It's orchestrated. Performances are always orchestrated. You think I call on you randomly, Samantha? It's all planned. I can assure you of that. Dora, just wait till Tuesday when you're right in this classroom with me. I cannot wait. Let's go home and pick up on Thursday.